The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, this is Jennifer Wilson here at the University of Illinois. I'm your hostess today for the Joint Centers webinar for January 2017. We have student presenters here from DeRISK, RESEAU, and the WIN Center, and I'm excited to go ahead and get started. We've got 3 presenters here today, and they will each share 15-minute presentations, as well as we'll have a 5-minute Q&A period. You're able to submit questions via the chat/questions box, and I can see those and read those to the presenters. Or if you would like to ask a question orally, just submit that in the chat and question box, and I will call on you and unmute your line. This webinar will be recorded and posted on drinkingwatercenters.net, as well as the Joint Centers YouTube page. Here's our lineup for today. We have Natalie Hull from DeRISK, Julie Bliss Mullen representing WINS, and Sean McBeath representing RESEAU. I'm going to go ahead and change the presentership off to Natalie so we can just jump right in. Unmuting Natalie. All right, welcome Natalie.
All right, thank you. So today I'm going to be sharing with you some research on UV disinfection, looking at the UV wavelength-specific response of MS2. So MS2 is a virus, and viruses are important for UV disinfection because in the United States, UV disinfection drinking water regulations are based on adenoviruses. Adenoviruses are human pathogens that cause gastrointestinal and respiratory illnesses. Adenoviruses are very resistant to the monochromatic emissions by low pressure mercury UV, but they're more susceptible to the polychromatic emissions by medium pressure UV. And here at the bottom I'm showing you the relative lamp energy for each of those types of UV. And relative lamp energy means that the emission for the lamp at each wavelength is normalized to the maximum emission. So to achieve compliance with regulations in the US, you have to demonstrate the equivalent of 4-log adenovirus inactivation. And this requires a low pressure UV dose of 186 millijoules per centimeter squared. And this can be just high enough to be prohibitive for small systems in some cases. With the polychromatic emissions, though, you can achieve the same level of inactivation at lower doses, and this has been shown for medium pressure UV. And we're starting to show this for wavelength-specific combinations of new UV sources like UV LEDs. And so to best optimize these wavelength-specific combinations of UV sources, we need to understand the spectral magnitude and mechanisms for how our pathogens and the surrogates that we use to model them are disinfected. MS2 coliphage is a commonly used surrogate for virus disinfection. On the right here, I'm showing you the action spectrum for both MS2 and adenovirus. The action spectrum means that the relative ability to disinfect at each wavelength is taken relative to 254, where low pressure UV emits. And so if you have a higher action, that means the microbe is more sensitive. So you can see both of these viruses are more sensitive to UV at wavelengths below 254. You can also see that MS2 has less relative sensitivity across the UV spectrum at those low wavelengths. So it's a conservative model for adenovirus. It's also non-pathogenic, and so it makes it easier to work with. And even though MS2 is much smaller than adenovirus and has a single-stranded RNA genome, while adenovirus has a double-stranded DNA genome, both of these viruses rely on proteins on the surface interacting with their host cells in order to initiate an infection. And thinking about both of these cell components, both the genome and the protein damage, helps us to best optimize the wavelength-specific disinfection. So we've known for a long time that genome damage is important for UV disinfection. It causes organisms to not be able to replicate. And the wavelengths that are important for damaging the genome are highlighted here in pink. The main area for targeting DNA absorbance has a relative peak around 260 nanometers, and it picks up again at wavelengths below 220 nanometers. We're starting to learn that protein damage is also important for disinfecting microbes, especially viruses. And you can see, so proteins are important for the structure and function of microbes. And the wavelengths where proteins absorb the greatest are below 240 nanometers, and then a secondary but smaller peak around 280 nanometers. And we can start to think about applying these wavelengths to optimize disinfection. So here again I'm showing you the action spectrum for adenovirus, but I'm also showing the action for damaging each of those components, the DNA damage in green and the protein damage in blue. So you can see the DNA damage is relatively constant across the UV spectrum, but protein damage is increased at wavelengths below 240 nanometers. And so this makes us think that proteins play a role in disinfection of adenovirus. And we want to think about how our surrogate microbe, MS2, is a good model for, or is not a good model for adenovirus. And we know that inactivation is a function of genome damage and protein damage for both of these organisms. And we want to think about how we can use what we know about the spectral response to inform wavelength selection for UV design and implementation. So to address these questions, I've done some experiments. The first, I measured MS2 inactivation after exposure to wavelengths targeted for genome and protein damage. So to target the genome, I exposed MS2 to a low-pressure mercury lamp emitting at 254 nanometers, and I'll be calling that L. To target the protein absorbance and protein damage, I exposed MS2 to a krypton chloride excimer lamp emitting at 222 nanometers, being supplied in a partnership with RESEAU, and I'll be calling that K. To simulate a polychromatic emission, I applied these UV sources together. The first way I applied them together was in a sequential exposure. So I first exposed the MS2 to the low pressure lamp and then to the excimer lamp to target genome damage and then the protein damage. I also exposed MS2 to these same sources but in reverse order. The second type of combination exposure I tested was a simultaneous exposure for MS2 to target both the genome damage and the protein damage at the same time. I'll be calling that L plus K. After UV exposure, I measured the concentration of MS2 in the samples by a combination of spot and pore plating to determine the plaque-forming units per mil, which tells you how many virus particles were able to infect the E. coli. I calculated the log reduction as the log of the control versus the UV-treated samples. Looking at these results, on the left I'm showing MS2 log reduction as a function of the total UV fluence, or the UV dose. And you can see the low pressure in blue has the lowest, the less steep dose response, and as we move up with the sequential exposures, KL and LK, we see a steeper dose response, and then the simultaneous L plus K is even steeper. And the most steep dose response is from the K, the excimer lamp, emitting at 222. And we can think about these to compare them in terms of the UV dose required to achieve a given log of inactivation of MS2. And you can see that the excimer lamp, the K UV source, has the lowest dose required to achieve a given log reduction. And the low pressure has the highest. And we can see that by adding a combination of the excimer lamp with the low pressure lamp that is traditionally used, we can achieve the same log reduction but with a lower UV dose. And this makes sense because we're targeting the MS2 at the place where the action is greater with the 222 nanometer, and then with the greatest potential for DNA damage at the 254 nanometers, and that helps us achieve more efficient disinfection by using a lower dose. To understand why this might be happening, I looked at the MS2 protein damage across the UV spectrum. I exposed MS2 to a single dose of UV. I used the excimer lamp again, the low pressure lamp again, and I also targeted other wavelengths using a filtered deuterium lamp and LEDs supplied by Aquasense, and you can see their relative emissions in the bottom plot. After UV exposure, I quantified the MS2 coat protein that makes up the shell of MS2 using gel electrophoresis and image densitometry, and calculated the damage as a ratio of the amount of protein before versus after UV treatment. And looking at these results, I'm showing you the action across the UV spectrum, and you can see at wavelengths below 250, we have a greater action for the coat protein damage, which shows that lower wavelengths cause more protein damage for MS2. And bringing this back and thinking about how MS2 compares to adenovirus, here's the action spectrum we've already looked at for inactivation of the two viruses. We see both of them are more efficiently inactivated at wavelengths below 250. And starting to look at why, Sarah Beck in the London lab did some research on the genome damage for both of these viruses. And you can see that for MS2, there's a high action for genome damage for wavelengths below 250, and then there's a relative peak for genome damage around 260 nanometers, which aligns with the peak absorbance of nucleic acids. However, for adenovirus, we don't see that increased action for genome damage at low wavelengths, but we do see that relative peak of genome damage at 260 nanometers. And this could be because adenovirus has a much thicker protein shell that could be protecting the inner genome from damage, whereas MS2 has a much thinner, more simple protein shell to protect the genome from damage. And finally, looking at the comparison in protein damage for these two viruses, you can see both viruses have enhanced protein damage at low wavelengths, which aligns with the spectral absorbance of proteins. To summarize, both of these viruses had genome damage with a relative peak around 260, which aligns with the DNA absorbance and RNA absorbance, and protein damage occurs efficiently at wavelengths below 240 nanometers. For MS2 only, we see genome damage at low wavelengths. And we can use this information to start to design UV reactors. Ideally, you would combine UV sources targeting the maximum protein absorbance and the maximum DNA absorbance. But if you're trying to design an LED reactor, there aren't LEDs readily available at those low wavelengths. So you can target the secondary peak of protein absorbance and still target DNA absorbance at the higher wavelengths. And this has been done by a company called Aquasense. They built the Pearl Aqua, which is the first certified UVC LED flow-through water disinfection system for drinking water. And we've been testing this in the London lab to prepare for field implementation at a small system in Colorado. And what I'm showing you here in the plot is the MS2 reduction equivalent dose from low pressure UV. So this is calculating the equivalent dose as if I had exposed these samples to low pressure UV, but for the flow-through testing. I tested various flow rates and various water qualities, and I'm plotting here each UV transmittance at 285 nanometers because that is the wavelength used by the flow-through reactor. And you can see that as flow rate increases, the UV dose decreases, and also as the UVT decreases, as the water becomes less clear, the UV dose decreases. And we can use these curves and this validation to help inform the flow rate that we can use. So a lot of utilities try and target 40 millijoules per centimeter squared. And you can see at that required dose, and for the UVTs that are typical of the field site, which range from 95 to 93, we would need a flow rate of about 0.5 liters per minute to target that dose. Just to summarize the big picture for this, both genome and protein damage may play a role in inactivation of both of these viruses, MS2 and adenovirus. And we can use these wavelength-specific data to drive UV source sensor and sleeve technology advancements. And these data and the implementation studies can support adoption of tailored wavelength technologies like UV LEDs to help small systems and just in general achieve required disinfection but using lower overall doses. Thank you.
All right, great. Does anyone have any questions for Natalie? I don't have anything submitted yet. I'm going to pop something in there real quick. Or we just move on to Julie here. If you do have a question for Natalie, just go ahead and submit that later and we can pop back at the end. All right. Julie, if you want to go ahead and share your screen.
Okay.
All right, looks great.
Great. So thanks for inviting me to speak today. My topic is relating ORP to chlorine concentrations for the ultimate use in in-home treatment systems sensing. So first I want to talk about point-of-use water treatment in general. So why in-home water treatment systems? In general, specifically in older areas like New England, U.S. infrastructure is failing. It seems to be, over time, it's leaking. It's causing chlorine residual problems at long water ages. And as a result, it's leaching pathogens from distribution systems. In some cases. And so ASCE has come up with an estimate of how much money it's going to take to repair this infrastructure. It's about $1.66 trillion, and they're estimating that this is going to more than double in the next decade. So we have a solution. The solution is in-home oxidation treatment systems. And so the reason we're looking at in-home oxidation systems is because we need to make sure that we can disinfect, specifically with these problems with infrastructure. So let's talk about in-home systems for a minute. The main in-home systems that we commonly use are adsorption systems and reverse osmosis systems. So you can see common in-home systems are point of use. So the faucet could be on the countertop, under the sink, also pitcher, and what we call point of entry, which would be in a basement. And so as you can see, most of these systems don't have some kind of sensing technology, and so they rely on the consumer to replace the filters, in which case almost all of those types of systems require filters. So there are a couple systems that do have sensors, in which case most of the time they're time-based sensors. You can see here Brita has done I guess a good job with their time-based sensors. You'll see change or change soon or good. But really, what does that mean? It just means that over time, so say it's, for example, 3 months, and I'm not exactly sure of that time, but about 3 months goes by and all of a sudden it says, oh, it beeps, you need to change it. But in fact, it doesn't actually take into account any water quality. They actually now have a sensor that you can touch now where Brita alerts Amazon, and then they'll send you a replacement, but that's just based on a time-based system. So the other ones, which are a lot more rare, are quality-based, water quality-based. So there are two examples of the water quality-based sensors that I have at the bottom of the screen. The first is Ecomo. They claim to monitor heavy metals, bacteria, pH, harmful chemicals, and other hazards. And so you can see here on their app that I guess they have used. So the homeowner can actually see those concentrations, and if there's a problem, I assume it beeps or something along those lines. And that's great, but actually it monitors so many things, including harmful chemicals and other hazards. And it seems relatively complex. And on the bottom right over here, that's a photo from a company called Me Water, and they use a total dissolved solids indicator or test for a water quality indicator, which could be helpful. But again, as going back, I think it's really important for there to be some kind of oxidation or disinfection sensor. So we started looking at sensors, and we came to realize that ORP, which is oxidation reduction potential, might be some kind of solution to in-home water quality sensors. So ORP measures the redox potential of everything in water. So it can, the redox reactions that are really important in drinking water are precipitation of iron and manganese, nitrification, corrosion of distribution system materials, and what we specifically are focusing on is microbial disinfection. So ORP is pH independent as opposed to chlorine monitoring. And what I mean by that is when you have an ORP scale down here, ORP, the more negative the scale or the reading, the more reducing the overall potential of the water is, and the more positive, the more oxidizing the water is. And so when you see an ORP reading, say it's 600 millivolts, for example, this is 600 millivolts regardless of pH and whatnot, as opposed to if you were receiving a chlorine measurement, say it was 1 milligram per liter, but in fact you don't really know what that 1 milligram per liter means, because we all know that there are certain forms of chlorine, and some have more oxidizing potential than others. For example, HOCl over OCl minus. And so ORP, we use in the lab multiple probes for ORP, but here are two. They're just small handheld ORP probes, and they are sort of similar to measuring pH, except for instead you're measuring the oxidizing or redox potential. We also have a couple of industrial sensors that are a lot larger, that can handle much larger flows. So the Nernst equation is what we use to model ORP. It's derived from thermodynamics and can be used to model ORP, but it's very complex. Although it seems simple, it can be extremely complex. The Nernst equation measures the energy of exchangeable electrons associated with a given half-cell reaction. So the Nernst equation is here. E sub h is the ORP of the sample relative to the standard hydrogen electrode. It's also called the redox potential. That's E sub h. And then you have the standard potential, and then from thermodynamics we have the universal gas constant, temperature, the number of moles of electrons transferred in a reaction, Faraday's constant, and then you also have the activity coefficient. So the activity coefficient is very similar to the equilibrium constant, and it can be simplified to the concentration of the reductants over the concentration of the oxidants. And so when you simplify at 25 degrees C, standard temperature and pressure, the Nernst equation can be simplified here. And so similar activity coefficients, or some activity coefficients for HOCl, for example, is 1.49 volts, and OCl minus is 0.9 volts, and you can see that difference of lower pH and how it has an influence in the standard potential. But what can be a little bit complicated, one part that can be complicated, is you have to account for this conversion factor. The conversion factor is really dependent based on the manufacturer of an ORP probe. So a lot of times they'll have these conversion factors for you. And for example, we actually use the Thermo Orion manual. We use the Thermo Orion ORP sensor, and so we can use these C values. And so when you do the calculation, it's the measured ORP is actually this equation down here. So it seems very simple, but it can be very, very complicated. ORP values are often normalized to the standard hydrogen electrode. They're easier to model with simulated water. They can be very difficult with natural water due to complex chemistry and the fact that the redox equilibrium is rarely achieved. So we do have some problems. So there are three objectives to this research. The first is to really get an understanding of the relationship between ORP and chlorine concentrations. And we're focusing on chlorine because chlorine is very commonly used in disinfection systems is potentially, it has a potential to be used in home systems as well, at least for mixed oxidant scenarios as well. We'd like to correlate the ORP and chlorine residual to formulate a model for monitoring oxidant residual in a water distribution system and also in the home as well. And we'd like to understand the electrode response and stability to different chlorine concentrations with varying flow rates. So at first we developed a method where we had water that was buffered to different pHs, and it was pumped through a manifold, through this manifold here, with different pH probes, different ORP probes, pH probe, and a couple others. And it was continually cycled through back into the reservoir. And every couple of minutes we would add more oxidant solution. In this case it was sodium hypochlorite because we were measuring free chlorine. So we would add the oxidant solution in here, pump it through, and then once we reached the ultimate chlorine concentration, we would then dilute it with buffered water and bring the concentration back down. So for analysis, we would take RAB samples for DBD colorimetric analysis, and we also ran the samples through UV-Vis spectrophotometer, and we looked at the absorbance of HOCl and OCl minus. So first we looked at these samples in a batch scenario, and that's over here. You can see that the ORP values increase sort of semi-linearly. You can see linear, sort of semi-linearly, and these trends are very similar with works from other literature. And you can see down here is pH 9, this was pH 7, and this is pH 5, just because we wanted to get a really broad range of pH. And based on the standard potentials, the ORP values follow the expected trend, where the higher the pH, the lower the ORP, and the lower the pH, the higher the ORP. So this sort of made sense, but we were just trying to get a gauge into what the relationship of ORP versus free chlorine would look like in a batch system. And then what we decided to do was run it through a flow-through system, as I described on the previous slide. We varied flow from 100 milliliters per minute up to 1 liter per minute. And we didn't find a whole lot of variation. In fact, they were almost exact. And so we chose a flow rate of 3 milliliters per minute, as it was for us to measure. And you can see the patterns of pH is sort of follow very similar patterns to the batch systems. So what we ended up doing is we added 0.1 milligrams per liter of sodium hypochlorite after every 5 minutes, because we figured at about, or we measured at about every 5 minutes the ORP probes would reach steady state, and so we knew that that was about the time that the ORP probes had completely responded. And so we went from concentrations from 0 milligrams per liter up to 1 milligram per liter, since that's really the range that we're concerned about at the end of a distribution system when there's not a whole lot of chlorine left over. What's really interesting is that we see kind of there isn't a whole lot of pattern of pH in the beginning. It starts with 6.2, it goes to 8.2, and then 7.2. This is really interesting, but one of the reasons why it might not follow this similar pattern as you would expect is because of the low ionic strength. You can see as time went by, as we increased the chlorine concentration, actually they did end up where the lower pH was the highest and the highest pH was the lowest. And actually the deviation in between these is very low, but we are pretty concerned about this area down here, and we plan to run more experiments in the future. So in terms of future work, we're developing a really large network of sensors and really manifolds. So we have different ORP sensors, different pH sensors. We have, I think, 2 chlorine amperometric sensors, SCAN, and 1 chlorine colorimetric sensor. And instead of having it be a flow-through system, we are pumping the water, same as we did before, through a pump, in through a manifold which diverts the flow rate to each of these systems here. We're going to have about a flow rate of 700 milliliters per minute, since that is a flow rate that all of them can handle. And we're hoping to optimize the retention times so that the water reaches the sensors around the same time. We're within a couple seconds. And all this data is going to go into data log, and we will be able to compare things like reproducibility, response time, etc. We're also hoping to be able to compare ORP versus the theoretical potential, although this is extremely complex, especially when you have more complex water qualities. So we'll repeat the experiments with different oxidants, for example, chloramines, ozone, and oxygen. We will also repeat them with tap water and mixed oxidant solutions. So our end goal is to really incorporate this ORP sensor into some kind of in-home water treatment system, and not so much of absorption or reverse osmosis, but again, we're really concerned about disinfection. And so we've kind of come up with a device that creates many different secondary oxidants. For example, it creates chlorine, it creates ozone, hydroxyl radicals, and other things that we have yet to measure. But I just wanted to show you what an ORP versus voltage, an ORP versus chlorine concentration shows. And in fact, I just want to clarify too, this is an electrochemical system, so it would be used for the home, plugged into the wall, or into an outlet. And there's an anode inside, in which case the oxidants are produced at the surface. And so we ran some experiments with this and the ORP probe, and we changed the voltage. And really, the voltage is just beside the power applied, it's an indicator of the amount of oxidants that are generated. And so it's a whole lot more complex than a semi-linear relationship. you can see that in this case, the ORP started around 600 and actually dropped really quickly down to the negatives and stayed in the negatives. And this is mimicked in the same way of free chlorine. So as voltage increased, the free chlorine increased, but the ORP decreased. And so this is a mixed oxidant solution, also with real water. This was tap water. And so what we're really trying to do is develop some kind of model so we can use not just deionized water, but real water, and try and incorporate into some kind of in-home treatment system like this. So we definitely have some work to do, but we're on our way to hopefully coming up with some sort of model. And that's all I have. So I just want to thank my advisor, Dr. Reckhout, and some staff in the lab, Patrick and Sherry, for their continuous help. So that's it. Thank you.
All right. Great, Julie. Thank you. We did have one question come in for Natalie, but what we'll do is we'll hold that till the end after Sean's presentation. And if you do have a question for Julie, go ahead and submit it as Sean gets started, and then we'll just use the time we have at the end for the questions. So you have time to think about it. All right. Julie, if you could hand the presentership over to Sean, that'd be great.
Okay.
All right, there you go, Sean. We can see your screen.
Perfect. Thanks, Jennifer. I'm happy to be following Julie's webinar up with another electrochemistry focused project. So I'll be talking about some of the work that RESEAU has been doing with electrocoagulation. So just jumping right into it. So just a brief overview and some motivation for our research. This is a map of Canada, and in each province you'll see a kettle, and each kettle is representing a drinking water advisory. The number below it represents how many of those drinking water advisories there are. And if you look at our home province here, where our school is located at that red star, you'll notice that we actually have by far the most boil water advisories than any other province. There's a few reasons for this, but one of the main reasons is just due to the remoteness of some of the communities. We have a lot of remote communities. Some are coastal communities, only accessible by boat or by plane. We have communities nestled into the Rocky Mountain ranges, and we have very variable weather. So when a community may be accessible in the summer, sometimes it's buried under snow in the winter, and this is taken near a city called Prince George in northern central British Columbia. And it's not that we don't have water. We have an abundance of water. The Fraser River runs through the middle of our province and then out through Vancouver, but not all of it is readily consumable, so it's very brownish in color. This is partly due to natural organic matter. So I'm not going to go over this slide in detail, but obviously there's a lot of unit operations that can pose as a challenge for these remote communities. And in particular, I'm going to talk about challenges associated with coagulation and flocculation. So a remote community will, its treatment, its water treatment will be dictated by a few things, and one of those things is the remoteness and accessibility, as I said. Can we get chemicals to the community, or is there trained operators? And of course the water quality will dictate what the treatment is.
Sean, I'm sorry to interrupt you, but you haven't clicked on any future slides yet? We're still on your first slide.
Oh, weird.
You might try going ahead. Yeah, we are seeing it change now.
Okay. Not when I full screen it? You're not seeing any change?
I am not seeing your full screen. I'm still seeing your slide sorter view. Okay. But when you do change it in the left sidebar, we can see that change.
Let me try this. Are you still seeing my screen?
I do still see your screen, yes. I don't see it full. There we go, full screen. I see remote community solution.
Perfect. All right. All right. So typical community, yeah, so you missed some really nice slides before, but I won't go back. So a typical rural community solution would be to just disinfect the water. And of course, communities that don't or can't do that are put on boil water advisory or drinking water advisories. And now we're knowing that if we just solely disinfect with chlorine and there is natural organic matter present in the water, we're risking exposure to disinfection byproducts. So this is where electrocoagulation comes in. So as I mentioned, that chemical supply chain poses a problem for remote communities because it's hard to supply those chemicals. That chemical supply chain is hindered. So electrocoagulation addresses this by generating coagulants in situ, so right in the water that you're treating. And how this works is using an electrochemical cell and a DC power supply or some external power supply, you provide current to the electrochemical cell. As current is provided, your iron anode or any other sacrificial anode like zinc or aluminum dissolves into the water as a metal cation, and then on your cathode side you have reduction of water into hydroxy anions as well as a hydrogen gas byproduct. When these metal cations and hydroxy anions meet in solution, they synthesize what are known as metal hydroxides, or more specifically in my case, iron hydroxides. And it's these iron hydroxides that function as coagulants. So with respect to iron, there could be up to 16 different species of iron hydroxides that could be formed, but we see more predominating species such as lepidocrosite, green rust, and magnetite, just based on the pHs that we work with, of course neutral pHs, dissolved oxygen, and potentials, electrical potential that is. So in previous research, electrocoagulation has been shown to be effective at removing fluorides, clays, heavy metals such as arsenic and mercury, as well as natural organic matter. It's good because it eliminates that chemical supply chain. Instead of supplying a community with chemicals, you're now synthesizing the chemicals on site. It's known as being operator friendly when designed properly, and it's an on-demand process. So what's yet to be uncovered is how it works at a large-scale operation. So currently the largest operation is 10,000 liters per day, which is about 7 liters per minute. However, this is a batch process. And it's a complex electrochemical process. That's to say that there's a lot of electrochemical variables that must be considered, that an engineer should consider when designing an electrocoagulation process. So with that in mind, our research objectives were to evaluate iron electrocoagulation as an alternative to traditional chemical coagulation for the removal of NOM. We did this in two parts. Our first part was scaling up the process ultimately to 10 liters per minute, first starting at 1.35 liters per minute and then 5 liters a minute in the lab, and then 10 liters a minute on site at a pilot plant. And in doing so, we want to understand the effects of current density, the inter-electrode gap, as well as metal loading. So I'll get into what those variables are in just a couple of slides. Our second part was to map the current density distribution along the electrode surface. We knew that mass transport of the water flowing through the reactor would have some influence on the current distribution on the electrode surface, and we want to see what these effects were, or confirm our suspicions. So today I'll be focusing on part one of our research, and more specifically our pilot scale work at 10 liters per minute. This is our electrocoagulation setup that we designed and built for all my experiments. How it works is water enters through the baffled inlet and then up and over into the electroactive area of the reactor, and then out a baffled exit, and then out the outlet tube. So this could accommodate flows of up to 10 liters per minute and had an electroactive area of 310 centimeters squared, and that's for one electrode. So this is a bird's eye view of our EC setup. You can see on the left side there it's a single cell setup, or what's known as a single cell setup, meaning one cathode and one anode. On the right side we have a 4-cell setup, so 4 cathodes and 4 anodes. And you'll notice that on either side, on the outside of the electrodes, there's acrylic flow blockers. This is put in place just so all the water is directed in between the electrodes. So I mentioned before the three parameters we were looking at were inter-electrode gap, and that's just the distance between the electrodes, the gap between each electrode. The current density is the absolute current provided to the system from the DC power supply divided by the electrode surface area. So if you remember the previous slide, if you have one cell arrangement and then you were to add it up or put it up to four cell arrangement, your current density would decrease because at the same amount of overall current, it's being spread out amongst more electrodes. Finally, metal loading is just purely the dose of coagulant, so how much coagulant you're adding to the system. And this slide's really just to outline how it can get pretty complicated pretty quickly. So as we change the current that we're providing the system, our current density is going to change and our metal loading is going to change. As current density changes and as the inter-electrode gap changes, we know that the electrical potential of the system is going to change just based on increased resistance. And also because of Ohm's law, we know that as current changes, potential is going to change. If you remember a few slides back, I was talking about speciation, iron speciation, and I mentioned that potential plays a large role on what chemical species or what iron hydroxide is being formed. And each iron hydroxide has a differing ability or affinity to remove a given contaminant. So one may be more effective at removing NOM than another species. So ultimately, it's the kind of coagulant that we're synthesizing as well as the amount of coagulant that we're synthesizing that's going to dictate how well our contaminant or NOM is being removed. So our scale-up process, again, metal loading, electrode gap, inter-electrode gap, and current density were our controlled operating variables. And our performance parameters were decreases or increases in DOC and UV absorbance to 254 nanometers. We first started our experiments at 1.35 liters per minute and then up to 5 liters a minute. And finally, we moved off-site and out of the lab and used real raw water at 10 liters a minute at a pilot plant. And it's these pilot plant results that we're going to be discussing today. So jumping into some results, our pilot plant was located on Texada Island off the coast of British Columbia, has a population of roughly 550 people. We were doing our work at Priest Lake Reservoir, which is the drinking water supply for the community of Van Anda on Texada Island. And it had an initial DOC of 6 ppm and UV absorbance of 254 of 0.13. And currently they only have chlorine disinfection and do have some concentrations of disinfection byproducts in their distribution system. On the left is our mobile treatment plant, and in the background is Priest Lake, and then on the right side there is my electrocoagulation setup inside of the pilot plant. So jumping into some results, looking at metal loading, effective metal loading, and current density. As you can see that as metal loading increases, both our DOC and UV absorbance decreases as we expected. So basically as the coagulant dose is increased, our NOM removal is seen to decrease. What we didn't see when factoring in or considering the error associated with each point is that we didn't see any significant differences with respect to current density. So as we added more cells, there is no significant difference for each metal loading. When looking at metal loading and inter-electrode gap, again, as metal loading increased, we saw continued decreases in DOC and UV absorbance. But when we were comparing inter-electrode gaps, we noticed that DOC decreased with decreasing inter-electrode gap, and the opposite phenomenon was happening with UV absorbance at 254 nanometers. And that is that we saw as the inter-electrode gap decreased, the UV absorbance actually increased. And this was, again, if you look at the error, it's not significant, these differences. However, this was suggesting that there was some effect on the molecular weight fractions of NOM removed with respect to inter-electrode gap. So to look into this further, we used high-performance size exclusion chromatography, and that we saw as smaller inter-electrode gaps preferentially removed smaller molecular weight fractions of NOM. So if you look at closer to 100 Daltons, you can see that 1 millimeter actually removed the most amount of NOM, whereas you go closer to 1,000 Daltons, it was actually the poorest performing inter-electrode gap. And these results confirmed what we were seeing with respect to DOC and UV absorbance. In general, we saw that larger molecular weight fractions of NOM are removed more efficiently with electrocoagulation, and this is a phenomenon that's been widely published in prior research, and that smaller molecular weight fractions were not effectively removed. So again, if you look closer to 100 Daltons, you can see that there isn't a large difference in removal, whereas if you look up by 1,000 Daltons, there's a great amount of reductions in NOM. Now, considering some energy requirements, and I'll highlight two of the conditions that were seen to have the highest reductions in both DOC and UV absorbance. So the energy is described by kilowatt hours per cubic meter of water treated, and you can see that the best conditions were operating at about 0.5 to 0.6 kilowatt hours per cubic meter of water treated. And using British Columbia industrial hydroelectric rates, this amounts to about 5 to 6 cents per cubic meter. When comparing it to an incumbent water treatment technology like reverse osmosis, it's about a quarter of the price. And when compared to traditional chemical coagulation, from what we've found is electrocoagulation is a little bit more expensive, but a lot of these studies don't take into account transportation cost of chemicals. And as we know, we're looking at electrocoagulation as a potential alternative treatment technology for remote communities. So if we were to consider the transportation of these chemical coagulants to a remote community, I think that we would see the price or the energy cost for both electrocoagulation and chemical coagulation to be very comparable. So to just summarize some of or all of the pilot scale results, at the highest metal loadings we saw average DOC reductions of 37% and as high as 43%. And we saw UV reductions of up to 55% on average for the highest metal loadings and as high as 56%. So some of you might think that doesn't seem like a lot of reductions for both DOC and UV absorbance. This was actually very challenging water to work with respect to electrocoagulation and chemical coagulation, because it had a very low initial specific UV absorbance, SUVA value, a very low SUVA value. So initially it was about 2.2, and we were able to reduce the SUVA to about 1.71, where there has been kind of a published stop point of 1.75, just based on the NOM and the fractions of the molecular weight fractions of NOM. We have concluded that although the absolute reduction of DOC was not a lot, we were reaching kind of the gold standard SUVA final values that we were hoping to get. We also saw that inter-electrode gap affect which molecular weight fractions were predominantly being removed, and that in general electrocoagulation preferentially removed larger fractions of NOM. We also saw that electrocoagulation required low energy, or had low energy demands compared to incumbent water treatment processes, as well as chemical coagulation. And just to quickly, one more slide on some of our related research. I mentioned current density mapping. So we used an in situ current density mapping process known as the partial electrode approach, which is used in the fuel cell industry. To understand how current was distributed along the electrode surface area. So in all previous electrocoagulation research, it's always assumed that your current density is uniform on the electrode surface, and we knew that this is probably not realized in reality, just due to different flow variations along the electrode surface. So using this technique, on the left side you can see the current distribution, and on the right side you can see some CFD models we did of our reactor, and it was really nice because our CFD models really matched our current distribution results well, and that areas that we saw that were predominated by high velocity water were also occupied by areas of higher current density. So this gave us a lot of insight into how current is distributed along the electrode surface. So some quick acknowledgements, RESEAU WaterNet for funding my research as well as all the other research, and my supervisors, Dr. Wilkinson and Dr. Massani. Thanks.
Thanks a lot, Sean. I'm going to take the presentership back here. We'll just making sure. Right. We do have one question here for Natalie. We also have a question of where we can find the slides later. I don't have the information on if the slides are being posted, but I know that the full recording will be posted on drinkingwatercenters.net. That's this URL here, as well as on the Joint Centers YouTube channel. I don't believe the whole slide presentations are being shared, but I'm sure the presenters would be happy to share those individually. All right, so we're going to go ahead and we have a comment question here for Sean from Chad. And Chad asks, can you comment on the comparative efficiencies between direct ferric coagulation versus EC dosing?
Yeah, actually I have a slide here. If you could let me share my screen. Okay, can you see the slide that says NOM removal comparison?
Yep.
So this work was done by a previous researcher in our lab, and he found that EC dosing with respect to traditional iron chemical coagulant dosing was very comparable. So as you can see that electrocoagulation is actually kind of nestled in between iron 3 sulfate and iron 2 sulfate, so very comparable in terms of DOC removal as well as UV absorbance removal.
All right, thank you. Any other questions, please go ahead and submit those in the chat/question box. I'm going to switch it over to Natalie in case she has any slides to share for her question. The question is, I was wondering how you calculated the UV dose for Excimer and UV LEDs. Sorry about that. I just unmuted Natalie. There you go.
All right, thank you. So to calculate the dose for these UV sources, we measure the incident irradiance at the peak wavelength for the emission, or the average wavelength if it's a polychromatic source. And then we correct that for the spectral response of our radiometer. We correct it for the UV absorbance of our sample. And yeah, we correct it for other factors that are specific to the experimental setup. But for the polychromatic sources that I use for the protein experiments, yeah, we calculated it the same way. And then for the sequential exposures, so to achieve a dose of 40, I used a dose of 20 for each exposure to get a total dose of 40. Does that answer the question?
I don't have anything else come in here, but I want to thank everyone for sharing. We have 2 more minutes here if anyone wants to submit any final questions or submit a comment to raise your hand if you want to ask an oral question. We can do that. Not seeing anything else come in here. So I want to thank everyone. I want to thank our presenters today for sharing a snapshot of their work. We'll have another Joint Centers webinar again in February, so watch for that information, and hopefully this recording will be posted within the next few days. I hope all of you have a great weekend.